My name is Takashi Angata, who works at the Academia Sinica, and it's my honor to chair this session at Advanced Course 1 on the glycan-binding proteins. And the first speaker, actually Professor Drickamer, will give us a brief overview of the lecture.
Thank you. Okay, so I have just one slide on the first presentation. Great, thank you. Just to tell you where we're going, we set ourselves the very modest goal of covering all of diversity, structure, recognition principles, and biology in 2 hours. So we'll do our best to do that, starting out particularly talking about the diversity of different sugar binding systems, which is what Maureen is going to talk about in the first lecture. In the second and third lectures, Matt Macauley and I will talk in more detail about two of the families, the siglec families and the C-type lectins. And that is obviously only a very small subset of the sugar binding proteins. In 2 hours, we could stand up here and just read names of sugar binding proteins and we wouldn't finish in 2 hours. So we had to select certain areas and we decided to focus on the mammalian proteins. There will be other opportunities to hear, particularly from Els van Dam and others, about some of the other lectins and their applications. But we'll talk about the human proteins and we'll start talking about diversity, which is what Maureen is going to cover. But we'll have a briefly introduction first.
Yes, let me briefly introduce Dr. Maureen Taylor, the speaker of the first lecture on the multiple families of animal lectins. Dr. Taylor started working on sugar-binding receptors such as mannose receptors of liver endothelial cells and macrophages as a graduate student at the Royal Flea Hospital in London. Her subsequent research at Columbia University, Oxford University, and Imperial College London has continued to focus on the related members of the C-type lectin family of glycan-binding proteins. Dr. Taylor's ongoing research is a joint project with Professor Drickamer. They use various techniques in the biochemistry, structural biology, molecular biology, and cell biology to gain an understanding of the molecular mechanisms underlying the recognition of glycoproteins and cell surfaces by glycan-binding proteins and the biological functions mediated by carbohydrate recognition. Of particular interest are sugar-binding receptors on cells of the immune system that recognize sugars found on pathogens and have roles in innate immunity, as well as receptors involved in glycoprotein homeostasis. They have also tried to make the field of glycobiology accessible to undergraduates and researchers from other disciplines by co-authoring the textbook called Introduction to Glycobiology, which has been translated to many languages. The title of Dr. Taylor's lecture is Multiple Families of Animal Lectins. So, ladies and gentlemen, let's welcome Dr. Taylor.
Thank you. Okay, so to start off, I'd like to just think briefly about the functions of the glycans that are attached to proteins and glycolipids. So we know that glycans are structural components of cell walls and extracellular matrix, but they can also modify the proteins to which they're attached. They can make them more soluble and they can stabilize them. But many functions of the glycans are actually not to do with the proteins that they're attached to. The glycans are actually encoding information which has to be recognized by other proteins. And so glycan-binding proteins, or lectins, are the receptors which recognize glycans and decode that information. And the lectins in animals have a variety of different functions. So extracellular lectins can recognize endogenous glycans, so those glycans that are on your own cells, or surface and secreted proteins, and they undertake functions such as cell-cell adhesion, Examples of these are the selectin cell adhesion molecules. They can be involved in cell signaling. The siglecs are one family of proteins which do this. They can do things like apoptosis. Many of you will have heard about the galectins recently. They can also be involved in endocytosis of circulating glycoproteins. So the acylglycoprotein receptor is a key protein that does this, clearing serum glycoproteins into the cell for degradation into the lysosomes. But intracellular glycan-binding proteins can also recognize endogenous glycans. So the glycans that are present on newly synthesized glycoproteins, these high mannose type structures, are recognized by calnexin and calreticulin, which take part in a quality control system for protein folding in the ER. And then there's another group of lectins, the M-type lectins, which mediate ERAD, endoplasmic reticulum-associated degradation, whereby terminally misfolded proteins are targeted for degradation. And this is through recognition of these types of glycans which are present on all of the newly synthesized glycoproteins. And then there are other intracellular lectins which mediate trafficking through the secretory pathway. So the L-type lectins, ergic 53, VIP 36, and then mannose 6-phosphate receptors which target a particular sugar modification, this phosphorylated mannose residue, and target lysosomal enzymes to the lysosomes. But lectins can also recognize sugars on foreign cells, on pathogens, and this mediates part of the immune system. And so many of these receptors are on macrophages. Particularly DC-sign, langerin, the mannose receptor are all cell surface receptors which can recognize sugars on pathogens and lead to them being either internalized and destroyed, which is a favorable outcome, but in some instances the outcome is unfavorable. So these sugar recognition proteins may allow intracellular pathogens to get into the cells. In this case, they're helping in infection. And there are also soluble proteins which recognize sugars on pathogens. So we classify lectins based on their structures into a number of different groups, and I'm going to tell you a bit about some of the largest groups. So the group that we work on particularly are the C-type lectins, and so one characteristic of lectins is that they all have a domain which is responsible for recognizing the sugars, and this is termed the carbohydrate recognition domain, or CRD. And so all of the C-type lectins contain a domain that looks like this, and in this case the sugar binding is calcium dependent. So the oligosaccharide ligates directly to a calcium ion and additional protein residues. And so the C-type lectins are the largest group of animal lectins, and you find the C-type carbohydrate recognition domain in association with a lot of other different types of domains. So some of the C-type lectins are soluble, And so these galectins have a collagen-like domain. You find the C-type CRD in proteoglycans with all these other types of domains. Other groups are membrane-bound. They can do things like endocytosis, cell adhesion, or signaling. And so the C-type carbohydrate recognition domain recognizes the sugar, decodes the information, and then these additional accessory domains determines what happens after those sugars have been recognized. The C-type lectins have a variety of different sugar specificities, either for galactose or mannose or more complex motifs. So the galectins, many of you will have heard a lot about. So the C-type lectins are all extracellular proteins. The galectins can be found inside cells in the cytoplasm and the nucleus, but they also do get outside of cells. And they don't have associated accessory domains apart from this one type, which has an N-terminal extension. And so this is what the galectin carbohydrate recognition domain looks like, has a beta sandwich structure. All of these bind beta-galactosides, and they either associate as dimers, so the single galectin CRD associates with an identical one to form this dimeric structure with the binding sites at opposite ends. Some of them actually have two galectin CRDs in a single polypeptide. So again, with two binding sites opposite each other. And so what these do typically is cross-link glycans. And that can lead to a variety of different functions, including cell signaling, autophagy, cell-cell adhesion, cell matrix adhesion. So they have slightly different specificities, but all of their ligands will contain beta-linked galactose. And so the next big group are the siglecs, sometimes called I-type lectins, because these all contain immunoglobulin-like domains. And you'll hear a lot more about these in the next talk, so I won't say very much about them. They're all membrane-bound receptors with one Ig domain at the end, which recognizes sialic acid. So this is the I-type CRD, as you can see here. And these other Ig domains are not involved in the sugar recognition. So just this domain at the end recognizes the sugar. And many of these have signaling motifs in their extracellular regions. So these are typically involved in cell adhesion and cell signaling. And then the last big group of lectins I'll tell you about are the R-type lectins. And this is a very interesting group because this is the only group which is found across different kingdoms. And so R-type lectins are named because they are structurally similar, to this domain in the plant toxin ricin, which recognizes galactose on cell surfaces, and that subunit causes the toxin to be internalized, and where the hydrolase domain can attack the ribosomal RNA. And so R-type lectins all contain a domain that looks like this domain in ricin, which is shown here. And so this has the typical beta trefoil structure, and this is the one from ricin showing it recognizing galactose. And so this typically binds galactose or N-acetyl galactosamine and its derivatives. And you find these in bacterial enzymes. So again, this domain is like a targeting domain which can bind the sugar, and then the hydrolase domain acts upon it. They're also found in the mammalian GalNAc transferases, which initiate O-linked glycosylation, where they're associated with the transferase domains, and probably what they're doing is, again, targeting the enzymes to a part of the polypeptide which has already had one GalNAc added. And so this domain can bind to the GalNAc, and then the transferase domain will add another one. And there are a few additional types of proteins that have these. One extracellular receptor also has one of these, the mannose receptor, and I'll tell you a bit more about that later. Okay, so we can look for these lectins in genomes, and this is a summary of major types of lectins in various genomes. So C. elegans, Drosophila, humans, and what you see is that the calnexin, calreticulin, the M-type and the L-type lectins, those that are involved in quality control in the secretory pathway, are conserved. But some of the later ones that are doing cell adhesion, cell signaling, are not found in these lower organisms. C-type lectins are found in C. elegans and Drosophila, but what's interesting is that these are different C-type lectins to the ones that are found in mammals. There are no orthologs between C. elegans, Drosophila, and humans. Siglecs are vertebrate specific. And one thing is that these model organisms, C. elegans and Drosophila, are really not useful for studying the functions of some of these major types of lectins. And so this is just an overview of the lectin diversity. If we look in animals, plants, and bacteria, we see that plants have a lot of different lectins which are not found in animals, and you can hear more about those if you go to Else Van Dam's talk on Sunday when she gets an award. But there are a couple of lectin families which are conserved in multicellular eukaryotes, and those are the ones involved in folding and intracellular trafficking. And the R-type is the only type of lectin that you find in all domains of life, so animals, plants, and bacteria. Okay, so let's look at a few examples of paradigms for animal lectin function. And apart from the mannose 6-phosphate receptors, I'm going to concentrate on the C-type lectins because those are the ones that you won't have heard much about otherwise. But let's just mention the trafficking of lysosomal enzymes because these were one of the first and best characterized types of lectin in terms of their function. And so these mediate targeting of lysosomal enzymes, which are synthesized along with all other glycoproteins in the ER, and then they have to travel through the Golgi, but they need to end up in the lysosomes and not outside of the cells. And so the lysosomal enzymes, their oligosaccharides are modified in by two different enzymes. An N-acetylglucosamine phosphotransferase adds an N-acetylglucosamine phosphate onto the mannose core, and then an N-acetylglucosaminidase takes off the N-acetylglucosamine to leave this mannose 6-phosphate marker. And this enzyme will only recognize parts of the lysosomal enzyme, not any other proteins. Okay, so that happens in the cis Golgi. And then in the trans Golgi, there are two receptors which can specifically bind that mannose 6-phosphate. And these are the mannose 6-phosphate receptors, or the P-type lectins. And so they bind the lysosomal enzyme in the trans Golgi network and traffic them to lysosomes and then to endosomes and then to lysosomes. And so there are two different receptors that do this. One just cycles between the trans Golgi network and the endosome. That one is the cation-dependent receptor. The cation-independent receptor can also go to the cell surface and recover any lysosomal enzymes which missed the receptors in the trans Golgi, and then mediate internalization of those back to the endosomes and then to the lysosomes. And so this is what the receptors look like. This is the cation-dependent one. You can see it's a transmembrane protein. It forms dimers. Its extracellular region contains the mannose 6-phosphate binding site, and you need this whole domain. And so this is the P-type carbohydrate recognition domain. And you can see the structure here and some details of the mannose 6-phosphate binding site, where the hydroxyl groups form hydrogen bonds with various residues in the protein, and the manganese group ligates the phosphate. The larger receptor has 15 domains in the extracellular region. So these all look like P-type carbohydrate recognition domains, but not all of them actually bind sugar. So 3 and 9 are the main two which can bind mannose 6-phosphate. And then there are two additional domains which also bind, but some of these bind mannose 6-phosphate with the N-acetylglucosamine still on it. And so that means it could capture any lysosomal enzymes that had missed the action of the N-acetylglucosaminidase. I should just quickly mention that there are 4 other proteins in mammals which contain domains which look like these P-type CRDs, and those are called MRH domains. So mannose 6-phosphate receptor homology domains, they don't bind, so they look like this, but they don't bind mannose 6-phosphate. And all 4 of those proteins are involved in early trafficking events. Okay, so let's turn to the C-type lectins. And a very well characterized group of these are the selectin cell adhesion molecules. And so these mediate adhesion of white blood cells to endothelial cells. And there are three different selectins. E and P selectin are on the vascular endothelial cells, and these bind to glycan ligands on white blood cells, neutrophils, and monocytes. And what these selectins do is this adhesion is not a very tight adhesion, so they mediate transient and weak interactions, which allow the white blood cells to slow down and come in contact with the endothelial cell, and that induces tighter protein-protein interactions, which eventually allows the white blood cell to squeeze through the endothelial cell layer to the underlying site of inflammation. So they mediate cell adhesion at sites of inflammation. And the other selectin, L-selectin, is actually on the white blood cells, particularly T and B lymphocytes, and it binds to glycan ligands on the special endothelial cells which line the entrance to the lymph nodes. So L-selectin does exactly the same thing in terms of slowing down the lymphocytes so that they can squeeze through the endothelial cell layer into the lymph nodes. And so this is very important for the targeting of the B and T lymphocytes from the circulation into the lymph nodes. And so the selectins all look like this. They have the C-type CRD on the extracellular region, and two other types of domains which are not involved in sugar binding. And they bind particularly to this Lewis X epitope, which has a sialic acid here and a fucose, and these Lewis X motifs are found on a lot of different glycoproteins, but there are particular glycoprotein ligands which are recognized by the selectins. So P-selectin glycoprotein ligand 1 for P-selectin and E-selectin ligand 1 for E-selectin. And this shows the details of the binding site, where the fucose ligates to calcium, the sialic acid is ligated, forming additional charge-charge interactions. Okay, so mannose binding protein in innate immunity. So this is a soluble lectin, which can mediate antibody-independent complement fixation. And so this binds sugars on pathogens, typically mannose and N-acetylglucosamine, which are common in bacterial cell walls and fungal walls. And so it binds to the pathogen, and then it has associated serine proteases which lead to activation of complement, giving you either cell lysis or opsonization. And this protein is definitely important in the innate immune system, particularly in young infants. And we know this because there are deficiencies in mannose binding protein which are linked to severe recurrent infections. And so this is what the mannose binding protein looks like. It has the C-terminal C-type CRDs which bind mannose, and collagen like domain, which allows it to associate first into trimers through a typical triple helix, and then into higher oligomers, and the associated serum proteases bind here. And these are the mutations which are associated with immunodeficiency. They don't affect sugar binding, but they affect the oligomerization of the protein and its ability to fix complement. So this is an example of one which is involved in pathogen recognition. And the final example I'll tell you about is the mannose receptor, which is on the surface of endothelial cells of the liver, and it's also on macrophages and dendritic cells. And this is a clearance receptor. And so you can see here, if you inject into an animal, a rat in this case, a glycoprotein which is terminated in N-acetyl glucosamine or mannose, it disappears very rapidly from the blood and goes into the liver. And it goes through the typical endocytic pathway, binding ligands at the cell surface, go into the endosome where the ligand is released, and then it's targeted for hydrolysis, and the receptor goes back to the cell surface. And this is a very interesting receptor because it has two types of carbohydrate recognition domain. So it has 8 C-type carbohydrate recognition domains which bind mannose. Only one of them seems to be very important. That's this one, number 4. And so the types of ligands which would have high mannose oligosaccharides on them are lysosomal enzymes. And so this protein binds released lysosomal enzymes that are released at sites of inflammation or tissue damage. You don't want them hanging about in the circulation, and this takes them out of circulation. It also has an R-type CRD at the end, and this binds 4-sulfated GalNAc, which is a particular modification which gets put onto pituitary hormones. And so this receptor is also responsible for regulating levels of hormones such as lutropin, which controls ovulation, or thyrotropin, which controls activity of the thyroid. And we've got details of the binding sites of both of these CRDs. Okay. It can also bind pathogens such as those you see on Leishmania, Candida albicans, or Mycobacterium tuberculosis. But this function is less well characterized. The lysosomal enzyme clearance function, we have very good evidence for that from the knockout mice. Okay, so I'll just leave you with a summary of different types of lectin families. I haven't had time to cover all of them, but I think you've got an idea of the overview and diversity now. Thank you.
Thank you very much. Is there anyone who wants to ask a question? Any question from the audience? Okay. May I ask one question, Dr. Taylor?
Yes.
So if I remember, recently there are some papers claiming that the calreticulin, which is expressed on the surface as a result of the the ER stress, and that's serving as some ligand for certain receptors being, and that's used for the recognition of these damaged cells. Do you think Well, apparently it's happening, but how much percentage of these theoretically may be participating in this kind of recognition event? And this may or may not involve the glycan recognition, actually.
Yeah, well, calreticulin definitely gets to the cell surface. As to how much, I'm not really sure, but it's definitely been reported at the cell surface and is proposed to have roles in internalizing proteins back into the cell.
Any questions from the audience? If you have a question, please use your microphone. Raise your hand and use a microphone. No? Okay. Well, if there's no other questions, then Dr. Taylor, thank you very much for your lecture. Next lecture is by Dr. Matthew Macauley from University of Alberta. Dr. Macauley started working in the field of glycobiology, chemistry and biology of gluconide as a graduate student at Simon Fraser University, and then he moved the field a little bit to study the siglec family of glycan recognition proteins as a postdoc at the Scripps Research Institute. He later obtained independent position at the Scripps Research Institute and then moved to the University of Alberta, where he continues his amazing work on the siglecs family of receptors. He has this amazing ability to show us the unforeseen possibility, open our eyes about the new possibilities of the function of the siglecs. And his laboratory continues to develop the innovative approaches to probe siglec-glycan interactions on cells and tissues, and to use the new insights about the biological ligands of siglecs to test hypotheses about the roles of siglecs in controlling immune cell function. The title of his lecture is, as you see, sialic acid binding immunomodulatory siglecs. Let's welcome Dr. Macauley.
Thanks, Takashi. Is this on? I don't think it's on. It's on? Okay. You can hear me? It doesn't sound on. All right, I'll go with it. Okay, thanks for the introduction. Takashi is also very known researcher in this field, so I actually feel funny giving this talk in front of him. So you can correct me if I say anything wrong, right? All right, so I'm going to tell you all about siglecs, so we'll do a little bit of a deeper dive than the last talk, but thank you, Maureen, for introducing siglecs. But I am still going to keep it pretty high level, so I prepared this talk with sort of the graduate student in mind that's maybe heard about siglecs, maybe you haven't even heard about them at all. So a little bit of historical perspective, and then a little bit of new stuff in terms of where the field is at. When do I get the ring, the bell? 5 minutes? Okay. Okay, so three questions I'm going to walk us through today. So what are siglecs? And then we'll talk about their ligands. Of course, they're glycan ligands. That's why we're all here in Taiwan for the glycans. So what do they do? What are they, and how do we study them? So those are the 3 subpoints I'm going to address there. And then I'll just end on some therapeutic opportunities, some things that are being pushed forward right now, and even into clinical aspects. So sort of some hot stuff that I'll introduce you to. Okay, so first point, which is what are siglecs? Well, we already got introduced to them, but a siglex stands for, it's just an acronym, of course, it stands for sialic acid binding immunoglobulin type lectin. And first thing here is that it's a type 1 single pass transmembrane protein. So that means that the single pass, nothing too fancy here. They have an N-terminal signal sequence. So for those of us that forget about that from our undergraduate, right, it's about a 20 amino acid sequence that's going to direct it to the ER. That's how they get out of the cell. And something that's we don't think about a lot even in the field is that most of these are glycoproteins themselves. Okay, lots of N-glycans on them. Some of them have O-glycans as well. Sort of an understudied area of how their glycans themselves affect their function. So sort of 3 parts, maybe 4, that I'll direct you as we walk through a map of a siglec. Of course, the real important part here, the VSET glycan-binding domain that's going to recognize the salic acid. Sorry, let's cut off a bit. We've got a series of C2 domains that are going to push this VSET domain out from the cell surface. A transmembrane segment, of course, whoops. And then the signaling motifs in the cytoplasmic tail. So we'll walk our way through what these are all doing. So the siglecs as a family, big family, Maureen already mentioned, 14 members. And I thought that was really cool, seeing Maureen's slide and showing basically they're absent from lower organisms than lower than invertebrates. So probably not a surprise, not a coincidence that those lower organisms don't really have salic acid either. Something for you to think about. Okay, so as I mentioned, they all have an N-terminal VSET domain, glycan-binding domain. They've got a really obvious from this slide is that they have a variable number of C2 domains that push them out a different amount from the cell surface. So, you know, the extreme examples are CD33 here, way down, and then siglec 1 that's pushed way up and sort of outside the cloud of the glycocalyx. And then in terms of the signaling motifs, I sort of classified them into 3 different types. The siglecs that sort of my lab, I guess we could say, cares about the most is those that have this little blue box, which is a motif that represses immune cell signaling. We'll get into that more. So that's contained in all these different siglecs. There's 2 here that you see don't have a motif. That doesn't mean that they can't do interesting things. They just don't have this interesting motif. And then these 3 on the end that have this positively charged residue in a membrane. If those of you that remember first year biochemistry, you know, a positively charged residue has really no business being in a transmembrane, and I'll touch on what that does. Okay, here's a nice slide that Jesu from my group, who's in the audience, first PhD student or graduate from the lab, put together, sort of summarizing where the siglecs are, and where they are is predominantly throughout the hemopoietic lineage. And so I'm not going to go through all these. These are in reviews, but you can see, I guess the thing I want you to take away is that each white blood cell type has a different expression pattern of all the siglecs. So, you know, from a simple expression pattern of one siglec to mast cells and neutrophils that have 4 or 5 different siglecs. So quite complicated with them all being expressed, but that means that there's lots of work to do still. Okay, another aspect that's talked a lot in the literature that is really, you know, spearheaded by Varkey is the conserved versus non-conserved subfamilies of the siglecs. So the so-called conserved and CD33 related siglecs. All this means really is that if you look at a sequence alignment of these 4 siglecs through basic vertebrates all the way up to humans, there's a very good conservation, whereas that this family there's not. Okay, that's all there is to it. In the laboratory, we know that a lot of us work with mice if we want to do in vivo experiments. So it's relevant for me to tell you how things are the same or different than mice. And the take-home message that you should think about, and take-home message is that they're really different. And it's something that us in the siglec field think about a lot and have to deal with. It's not always straightforward translating what we find in humans to mice and mice in humans. And here it is again. So this is taken from one of Takashi's reviews, sort of highlighting how the different members from rodents and humans are orthologs or functional orthologs of each other. And you can see that the pattern is quite complex. So for example, well, I'm not going to go through, but it's quite complex in terms of who's an ortholog of whom. Again, the take-home message is that when thinking about siglecs and trying to think about how results in mice might translate to humans and vice versa, you have to think about it and consider it. It's not always straightforward. Okay, let's go a little bit deeper now into the structure of siglecs. I think this may have been almost the same slide that, that Maureen had. This is from one of Jim's reviews 4 years ago. So this is the structure of siglec 2, or CD22, with 2 of its underlying C2 domains. This was solved by a colleague of mine in Canada, Jean-Philippe Julien. And you can see beautifully here it's an extended structure. One thing that I didn't put down in the notes, but not talked about a lot, but really is quite important, is that a lot of these domains have interdomain disulfide bonds between them that kind of stitch these domains together. And in fact, when we mutate those, we have very profound effects on glycan-binding. It just messes up the protein fold. Okay, so here we have the VSET domain, and I'll go into the VSET domain a little bit more on the next slide, but here you have beautiful interactions with the salic acid. The key interaction that I'll tell you about is this salt bridge with a conserved arginine. Okay, and so this is really what holds this together. This is not a strong interaction for those of us that are used to thinking in biochemical terms. These interactions for the siglecs range anywhere from 5 millimolar, are some of the weakest ones that have been measured. You'd be hard pressed to measure anything that's weaker than that, down to about 50 micromolar. These are natural interactions. So here's the entire siglec family. This was a structural survey that we took 2 or 3 years ago. We can add siglec 15 to this now. That structure was published 2 or 3 months ago. What I want you to take away from the structure of the VSET domain is 2 things. It's anti-parallel beta sheets, and it basically looks like a cup of your hand. So if you just cup your hand, that's where the salic acid is binding, right in the cup. That is not a deep groove. Okay. In my PhD, I studied glycosidases that tended to have a very deep hole where the substrate would go and react. These are very, very shallow clefts. If you talk to any pharmaceutical, anyone from a pharmaceutical company, they'll look at this and think that basically looks like the wall on a building. There's not a lot of grooves to exploit there. If we zoom in, we can again see here is the salic acid, and there's that critical arginine residue interacting with the carboxyl group on salic acid. Okay, just a few more sort of general ideas about the family is that there's these paired, 2 sets of pairs. This is something that Takashi is a real expert at. I only sort of know of. Siglec 5 and 14 are pairs, and by this we mean that the outer domains are like gene duplications. Their amino acid sequence is almost identical. However, 5 and 14, just staying with that one, you see that their cytoplasmic tail and transmembrane domain are very different. I'll get into that with the implications of that in a second. And same for 11 and 16. So, what is with these positively charged residues? So this is taken from an old review by Louis Lanier, a really classic immunologist, showing how these immune receptors can pair with negatively charged adapter molecules. These are some of the common ones that are expressed on immune cells, DAP10, 12, and Fc epsilon R1 gamma. So these have an aspartic acid residue basically positioned at the right location in the transmembrane to pair with the positively charged. So they're absolutely required. And not only are they required to stabilize that siglecs 14, 15, and 16 on the membrane, not only are they required to stabilize it, they also contain motifs of their own. And I haven't walked you through what these motifs do yet, but you can see that these are called an ITAM. Siglecs have an ITIM, and you might guess that these are activatory siglecs. Most siglecs are inhibitory. So very, very different. We call these activating siglecs, whereas the rest of them are inhibiting. Okay, so that's a very profound difference. Okay, so Yeah, so now I'm going to tell you a little bit about what these motifs do. So this is going back to this image. We've got glycan binding on the outside of this cell through the V-set domain and immune cell signaling regulation through the cytoplasmic tail. So what happens to this ITIM? How does it do its job? So under a certain physiological condition or a circumstance, and what that circumstance is really, what drives my lab, when would this happen? Which is, when does the siglec get phosphorylated? I'll touch on this in a little bit, when it might. But when it does get phosphorylated, that's going to create a docking site for a series of phosphatases that basically recognize that phosphorylated ITIM. So it's going to recruit a phosphatase to it, and then that phosphatase is going to be specialized at dephosphorylating other signaling components. So in very simple terms, this is basically going to dampen other signaling pathways. It's like a brake on the immune system. So there's some ideas out there that siglecs signal. I don't really think about siglecs that way. Siglecs don't naturally signal. They just modulate other signaling pathways, if that makes sense, right? If we cluster together some siglecs, that's not really expected to drive a signaling cascade like we're used to thinking, okay? So it's really about where and when this phosphatase is going to be recruited. Okay, so recruiting of this phosphatase for the inhibitory siglecs is going to inhibit other activatory receptors, and it's all about the location. Recruitment of the phosphatase needs to be close to the activatory receptor. That's what I'm trying to get across here. So, why is that? Why does it need to be close? Two things. The first thing I think is more obvious, which is the phosphatase just needs to be next to the signaling pathways that are happening. So, for example, we could call this an immune activatory pathway that recognizes, a pathogen, for example. So the phosphatase needs to be close to where the immune cell signaling is happening. It's just a location thing. The second one, though, is really not so obvious. I think this is something that a lot of people don't think about as much. And I'll just go back and to highlight the point, but this needs to be phosphorylated. The siglec needs to be phosphorylated. And it's not talked about a lot, or it's not studied a lot. It's hard to study because it becomes a chicken or an egg situation on who's doing the phosphorylation. So who's doing the phosphorylation is a family of kinases called Src kinases. The best characterized one in terms of phosphorylating siglecs is this Lin kinase, although there are other ones that are likely redundant. So these kinases hang out in activatory rafts where activation receptors are inducing immune cell signaling. So this is kind of profound. It's the same kinase that turns on signaling that then phosphorylates the siglec and turns it off. Okay? So that's the second reason why the siglec needs to be in proximity, because it needs to get phosphorylated. It then needs to recruit the SHP1 or SHP2 phosphatases. Okay, so it's all about location. And if we keep that sort of view in mind, that can help guide our thinking about how glycans may control where the siglec is, and in particular, control its proximity to the receptors that it regulates. And that sort of brings me to the second point. So siglec ligands, probably the deepest we'll go here. Okay, so just to remind you, it's all about location. It needs to be close. And we're going to look at how interactions with sialic acid containing glycans could influence this proximity. So to lay the groundwork, there's two types of ligands that have been discussed and talked about since the 90s. And that is a cis interaction and a trans interaction, basically just meaning an interaction on the same surface or an interaction with a glycoprotein on another cell, another particle. That would be a trans interaction. So here's how my laboratory sort of thinks about these interactions. We don't yet know how generalizable they are to all the siglecs. I'll leave you with that caveat. There's still a book being written for all 14 members on how they function. But some of these points are taken away through a few examples where we do know a lot about. So, in general, interactions with cis ligands keep the siglec away from the receptor that they regulate. Okay? Again, that's a bit of a generalization, and there may be exceptions to that, but this is one way to think about how siglecs function. Okay? An interaction in trans with another particle, another cell containing a ligand will naturally bring the siglec together with a receptor in the context of an immunological synapse. Okay? So they're just kind of forcing the two receptors together. And remember, proximity. Okay? So in this paradigm, we can think of cis interactions as keeping away and trans interactions as bringing together. Okay? So one way we think about this is that the cis ligand interactions are keeping the siglec away so that it can be ligated to its receptor when it needs to. When it needs to. So, when would it need to? That's the big question then. So, I'll give you two, Kurt mentioned sort of, historical perspective. I don't think I bring a lot of historical perspective here, but here's one. So here's sort of the older concept on, if you read papers going back to the 90s, on how siglecs function. They would describe siglecs as setting a threshold for activation. Okay? That was sort of the words that were used, and that still may be true, but in this model, basically, you have an activatory receptor that may have ligands for siglecs, which would be bringing it close together. And so in this model, the cis interactions would be continuously shutting down that receptor and suppressing it. Okay? For myself, I think of this as sort of an older concept, although it still may apply to some siglecs. This is sort of a newer concept that's that we're working on, which is it's trans interactions that are going to bring it together. I've already sort of introduced how that would be. And so the when is really critical. When would a trans interaction drive a siglec with an activatory receptor? Well, it would be in the context of recognizing sialic acid on another cell. So what cells have sialic acid on them? That's an important question to ask. And the answer is that, of course, a lot of lower organisms don't have sialic acid. So it's not really the fact that these organisms don't have sialic acid, it's the fact that our cells are really covered in a dense layer of sialic acid, right? One of a key characterization of higher eukaryotes. So that's why we call this non-self discrimination. So the siglecs are there basically to prevent an undesired or an unwanted immune response. Okay, I think I'm going to go through this kind of fast. There's lots of different models that this could, how this could be playing out, but I can direct you to this review. I'd rather skip forward so we don't run out of time here. Okay, so siglec ligands, sort of digging and drilling in deeper into what they are, not just sialic acid. Yes, they're all sialic acid containing glycoproteins or glycolipids, but exactly what are they? So first of all, a lot of us are familiar with this from the glycobiology textbook. Three main categories of sialic acid containing glycoconjugates: N-glycans, O-glycans, and glycolipids. There are a few other ones here that have sialic acid, but I'll just be focusing on the major class here, these three. And so all three of these have potential, and all three have been shown to be ligands for siglecs. So this is sort of taking it back a level for those of you that don't really know the structure of sialic acid that well. So there's a 9-carbon sugar here. I've numbered them here. And in mammals, there's three different linkage: alpha 2 6, alpha 2 3, and alpha 2 8 linkage. And here's the nomenclature on how those look down below. So siglecs, this was one of the first things that was described all the way back in the 90s. It was realized that siglecs have specificity in terms of the linkage they recognize. And for most siglecs, this has been I would say fairly well established at this point. And you can kind of go through each of these and see that each one of them has a slight preference for the linkage. Some are very strict and some are a little bit sloppier, but overall they have preferences. Okay, I would say in the last, by about 5 or 10 years ago, this was fairly well established. Now where the field is at is kind of digging deeper and saying, okay, we know that, for example, siglec 8 likes a 2,3 ligand. Yeah, but where? Where is it presented? And what other structural features of the glycan make that a siglec 8 ligand? Okay, so I'm gonna walk you through 3 things, and this is really, like my title says, this is work in progress, right? This is, the field is really writing the book on this now, and a few of the examples I'm going to show in a couple slides are work that's been done only 2 or 3 years ago. So the first thing is the type of glycan that it's presented from. Okay, so really well established examples of this: N-glycans, CD22, O-glycans, siglec 7, and glycolipids. I put siglec 6, but we had one paper on that, so I don't think it's that established. But there are other siglecs that bind to glycolipids. So where the salic acid is presenting from absolutely does make an impact on how specific it is for a siglec. Okay, what else? Well, the underlying motif, sort of where it's presented from. So, for example, the N-glycan branch, there's, I think, 2 or 3 papers that have shown that CD22 prefers salic acid, alpha 2,6 linked salic acid presented from the 1,3 branch on a biantennar N-glycan. So it prefers the 1,3 rather than the 1,6 branch. Alrighty. Another is that a siglec might prefer a disilyl core 1 motif for siglec 7. And for I had glycolipids. I didn't complete this one. So siglec 7 requires 2 salic acids off of a glycolipid. Okay, so there's more than just the linkage. That's the point. And then the last thing here is that there's specific proteins now. And these are all examples within the last 5 or 10 years. Specific proteins that bear silated glycoconjugates that are ligands for certain siglecs. Oops, I forgot to fill these in. Sorry. But again, the book is still being written, and I'm sort of leaving you with how does a specific protein, serve as a ligand for a siglec? Well, the answer is we don't really know, but we want to find out. And I think sort of that's where the field is at. It could be the density of the glycans on the protein, the abundance of that protein, unique glycoforms on that protein, or maybe there's some protein-protein interactions that are contributing as well. This is kind of, I think, where the field is going. So how do we study these interactions? I'll just kind of breeze through this. I think it's fairly straightforward. Glycan microarrays have been around for 20, 25 years, maybe 20 years. And still most of the knowledge that we've gained about siglec specificity comes through them. My lab uses a lot of cell-based assays. I won't go through that, but these are just complementary, not as high throughput. And these leverage soluble versions of the siglecs. Again, I won't go through these, but these are something that the field uses a lot. Siglecs are transmembrane proteins, and so difficult to work with, so soluble proteins ease that. And then 3 papers that came out only 2 years ago, I just wanted to draw your attention to, that have used cell-based arrays to make new inferences into siglecs, which I think are really powerful. They're definitely going to be used more in the future. So work from Henrik Klassen's group who's in the audience. Carolina Bertozzi's work on CRISPR screens, and then some of our work where we induce the expression of protein. So all sort of different yet complementary approaches. And really a lot of new insights into siglecs specificity have come from these. Okay, so I'll just kind of breeze through these case studies, but essentially I've told you that a lot of the siglecs are inhibitory, so they're a brake. And so the question is, in terms of trying to target these receptors, when would we want to target them? So in cancer and in dampening immunity. So here's sort of the idea here, is that when an immune response or cell is underactive, if we can inhibit an inhibitor, that's what a siglec is, most of the siglecs. If we inhibit an inhibitor, we're going to get a positive signal, okay? And so the idea of that is in cancer. Or when an immune cell is overactive, if we can put a brake on that, targeted dampening could be very effective and be a lot better than wholesale immune suppression. Okay, so these are two areas. I don't think I have time to go all into them like I wanted to, but these are two areas that have been moved forward in the last decade, and now there's companies working on both aspects of these. So I think there are opportunities and things to be looking out for. So just very, very, very briefly, I'll just breeze through this. So cancer upregulates salic acid on the surface. Siglecs are exploiting that, suppressing immune cells, and essentially here there's been a lot of activity in trying to then block siglec glycan interactions. The most advanced of which are Palion's technology of using neuraminidase to rip salic acid off the cell surface. Okay? And then in terms of the opposite, so that's in cancer, removing siglec ligands is going to increase an immune response. The opposite is when you would want to decrease an immune response, and that would be in the case of, for example, allergies, autoimmunity, and this is something that Jim Paulson's lab, which I was fortunate to be a part of, has really moved forward, and I'm not going to get into it, but here I'll mention that these are really great opportunities for dampening immune response. We all know that allergies is where our immune system is overactive, autoimmunity, transfusion, biotherapeutics, all places where if you could strategically knock down immunity, these would be useful. And so we've applied siglec ligands to selectively dampen immunity in these examples. So I'll let you look through those if you're interested. Okay, so happy to take some questions. If any of this sort of tickles your fancy, come talk to me about it after. There's lots. Edmonton's not always this cold. Sometimes it is, but yeah. Anyways, happy to take questions.
Thank you very much. I usually spend 2 hours talking about the same topic, and you did much better in such a short time. So is there any question? Could you please raise your hand and use a microphone?
Okay. Amazing call. Thanks for your presentation. So I would like to say some people will use the synthesized glycans such as microbeads or lipid anchor form to address the function of this siglec protein. So, but however, as you mentioned, there are some preference for the siglec binding, such as CD2 prefer for the N-glycan. So if we need to use this assay, what would we need to take in consider or some limitation?
I kind of missed the first part.
Okay. Sorry?
Do you want to just repeat the first part of the question?
Okay. So because some people will use the synthetic glycan for address the siglec, but as you mentioned, that some siglec will some preference for the N-glycan or other structure. So
you're asking about using synthetic glycans to try and.
Or some concern or limitation for this address.
No, there's many applications for using synthetic glycans. Unfortunately, I had to breeze through them at the end, but that's a big field that many contribute to, and using synthetic glycans, particularly with artificial chemical modifications to make them specific and high affinity towards a single siglec, is something that it continues to be very useful.
Thanks.
You mentioned siglec has some preference for glycan and glycan and so forth. So among these glycans, is there a preference for siglet to bind this N or O glycan made by particular cellular transferases?
Yes, absolutely. I would probably, some of the best examples of that are in the Boole paper by Clausen and Naramatsu, where they knocked out glycosylation pathways and in conjunction with that different sal transferases, and you can see very profound differences on siglec binding for certain siglecs. So the answer, I think, is absolutely yes. There's a lot more specificity there than I probably realized maybe 5 years ago.
Thanks. I have a question about, thanks your wonderful talk here. Yeah. So I have a question about the paired siglecs. So you mentioned that they almost have identical extracellular domain. So when they meet the same ligands, how the cells differentiate, how to respond to the same ligands?
Yeah, thanks for the question. Sorry I didn't have time to get into that. You should really take a look at Takashi's work. He's the expert on that. But briefly, if you had a cell that expressed both at the same level, well then the expectation would be that those signals would cancel each other out. And there are a few papers on that by Varkey. But there's polymorphisms in the population that some people only express one or the other. And so they're missing one of those signals. And that can make them more susceptible to certain diseases. So that's where sort of you really see this come into action. But I think what you're thinking is these are just going to cancel each other out, and then so the answer is yes if a cell expresses both. Some cells only express one or the other, so they can be at the transcriptional level, they can be differentially regulated.
Yeah. I wonder whether it's possible to compete the function of siglecs with free sialic acid, because there are some therapies where sialic acid is used as drug, let's say like this, in diseases. Do sialic acid interfere with the, let's say, function of siglecs?
Basically, no.
Okay.
Siglecs perform their function through engaging in multivalency and avidity, and this means tens, maybe hundreds of interactions simultaneously being engaged in an immunological synapse. And so soluble sialic acid has affinity of 1 millimolar, is not going to be able to outcompete that. There are a select number of examples where people have used sort of low micromolar, nanomolar synthetic ligands in a soluble form and been able to elicit an effect. But there's only a handful of those examples.
You showed that the siglecs have a variable number of C2 domains, and I was wondering, does it also impact the function of the siglecs? Is there anything known?
Yeah, not a lot has been done in that. Paul Crocker, who is I still think like the godfather of the field, he's the one that should be giving this talk, has a really nice paper out just, I don't know, not that long ago, where they took siglec 1, that's like super long, and then they chopped it down. And basically what that does is that the VSET domain is now in the glycocalyx instead of poking its head over the glycocalyx. And so there, on these shorter ones, now they start to bind in cis, and they can't bind in trans as much. So that's where this can have a big impact.
Thanks, Dr. Mateo. I have a question about the animal and also human siglecs. So when we talk, for example, siglec E, and which is considered the protein homologue for siglec 7 and siglec 9. So when we study the interaction with any siglec ligand, we can find always a difference between siglec E and always the affinity with siglec 9 and also 7. So how can we overcome this problem when we design some siglec ligands?
The ligands is, when we try to design synthetic ligands, you really have to go for a certain siglex. So there's not too many examples where it'll cross-react with a CD33 related member. So if your goal is to make a synthetic ligand that binds to siglec E, chances are I would bet that it wouldn't bind to siglec 7 and 9. There's too much dissimilarity between those. So that's not a big concern. Is that answering your question? Okay, we can talk more if you want more.
Okay.
Any other question from the audience?
Hi, Matt. I would just like to ask, how do siglec differentiate? Because we know that when virus is inside our body, they put on siglec on their surface or other pathogens. So how do siglecs differentiate those sialic acids on those pathogens compared to what we have in our body?
Yep, great question. So viruses can exploit siglecs. There's lots of really good examples of that in the literature. One thing is that siglecs are endocytic receptors. And so if there's certain siglecs on macrophages, for example, siglec 1 that we were just talking about, that can capture and internalize virus, and you might think, well, that's great for sort of immunity, but viruses can actually use that mechanism to go in and hide out within macrophages and then pop out when they need to come out. So the answer is that viruses are pretty clever, and they're probably putting on sialic acid to exploit siglecs to some degree. It's a war out there.
Okay. Any other questions?
Ku has one.
Okay, please go ahead.
Hi, Matt. Can you comment a bit on this issue that I think both Maureen and you mentioned that in C. elegans there is no siglec, and sialic acid in in Drosophila. Sialic acid in Drosophila is somewhat controversial. They certainly do have salar-transferase. So, and then another related question is that, is anybody know anything or study anything about siglecs in, let's say, fish, or even like the favorite zebrafish model?
I'm not the best one to ask about fish. Maureen, do you know offhand? I think there are some in fish, but I can't say definitively, so I should just not say anything. But Drosophila, yeah, whenever I see Vlad Panin, who studies sialic acid in Drosophila, I always ask him what the latest update on sialic acid in Drosophila is, because, yeah, like you said, it's controversial. The cell transferases are there. I think the donor's there, but where sialic acid is still like not super clear to me.
There.
But there's Maureen clearly showed that there's none. Thanks for all the questions.
Let's thank Dr. Macauley. Okay, so we shall move on to the last lecture of this session. That will be given by Professor Drickamer from the Imperial College London. Dr. Drickamer began his working in the field of glycan-binding receptors as a postdoctoral fellow at Duke University. These receptors have been the focus of his subsequent research as an independent researcher at Cold Spring Harbor Laboratory, the University of Chicago, Columbia University, Oxford University, and currently at Imperial College London. His work on identifying a common carbohydrate recognition led to the definition of C-type lectins as one of the major groups of glycan-binding receptors, and he's a godfather of C-type lectins, so to speak. His ongoing work is joint project with Professor Maureen Taylor, as I mentioned earlier, and their work is to aiming to develop a broad understanding of the biological roles of sugar recognition, and they have used a combination of biochemical, biophysical, etc. Approaches to understand how carbohydrate recognition domains provide selective recognition of glycoproteins and cell surfaces. The title of Professor Drickamer's lecture is How Structure Lead to Functions of the C-type lectins. Let's welcome Professor Drickamer.
Okay, thank you very much for the introduction and for keeping the session organized. So I'm going to cover the topics that are listed on the first slide here. Roughly speaking, talking about things we know pretty well, that is how the C-type lectins bind to sugars and selectively bind to the correct sugars to be useful. And then I'll try to touch on where we would like to be, but perhaps aren't quite where we would like to be in terms of understanding how that results in the functions. And Maureen has introduced you to the functions, so you can sort of see where we're headed. And I should mention that all of the kind of structural work underlying the understanding of how the proteins work is a collaboration with Bill Weiss and Hadar Feinberg at Stanford University, and we will keep throwing in results from glycan array analysis to understand what sugars different receptors bind to, and that is largely a collaboration with David Smith at Emory University. So I won't keep mentioning them, but they're there in all of the subsequent work. So we'll build up to the full binding specificity in stages, because the C-type lectins are characterized by one common feature, which is the way that they interact with monosaccharide ligands. And that common feature is that the monosaccharide interacts with the calcium ion, hence the C and C-type lectins. And that interaction involves the coordination geometry of the calcium. The properties of the orbitals on calcium just work out in a way that makes them very good for interacting with adjacent hydroxyl groups in specific orientation. And many of the ligands for the calcium are provided by the amino acid residues in the protein, but two of the ligands come from the adjacent hydroxyl groups on the sugar. But that's a pretty flimsy binding site. There's not much to it. Rather remarkable, in fact, that it is able to bind to sugar. Neither specific nor high affinity at that stage. But it works, because that is all it takes to bind to some of the C-type lectins, in particular the mannose binding protein. That's the only real interaction between the sugar and the protein, is through this coordination to the calcium geometry. So it's a very simple binding site, but it already does give you some selectivity, because the geometry requires that those two hydroxyl groups, in this case, be in the equatorial positions. And amongst the naturally occurring sugars in biological systems, that means primarily mannose, which is shown here, or N-acetylglucosamine, which would differ at the 2 position, and therefore that doesn't matter. So it can bind either of those two ligands, which vaguely tend to be more common on microorganisms, as opposed to things like galactose and sialic acid that are common on your own sugars. So you can start to see the beginning of a differentiation, even with such a very simple binding site. Now, the typical ligand, of course, is not just a monosaccharide. It's a monosaccharide in an oligosaccharide, such as this high mannose oligosaccharide. But when you see the structure, you'll see that, in fact, it is only one of the sugars that's interacting with this binding site, in the case of mannose binding protein. So you can go quite a ways with just that basic binding site. And in order to build that specific binding site, you need certain amino acid residues. And that set of residues includes the residues that ligate to the calcium. It includes some disulfide bonds, and it includes a series of hydrophobic aromatic residues that make the fold. And the useful thing about that is that we can recognize a motif that's going to be conserved in these types of domains, so that we can then look for that motif. Now something that's done completely automated. The Protein Data Bank does it automatically when anybody deposits a sequence and says, oh, that looks like a C-type carbohydrate recognition domain. So we can recognize them from the sequences and potentially start to make predictions. But we have to be very careful at this stage to distinguish the fact that there are whole families, many examples of domains that contain all of these residues but do not contain the calcium binding site. And those residues, from what I've told you, are not likely to bind, those domains are not likely to bind sugars. And so we make this distinction between the carbohydrate recognition domains, the CRDs, and the C-type lectin-like domains that would have a similar fold, but we would not make a sugar binding distinction. And so unfortunately we get lots of interest from people who say, oh, we have a domain that comes up with the C-type lectin domain. Can you help us study sugar binding? And we have to sort of tell them it probably doesn't, and we probably can't help you. Okay, finally on the simple sugar binding site, we can build a site that's very slightly modified from the mannose binding site and turn it into a galactose binding site. And by very slight shifts in the ligands in the protein around the calcium, we can make it prefer galactose, which has an axial and an equatorial hydroxyl group, instead of mannose with the two equatorial ligands. In addition, galactose binding sites are often characterized by a hydrophobic packing against a tryptophan residue. So that gives us an ability to make some crude predictions about sugar binding from simple sequence analysis. So at that point, we can start screening sequences and saying where are they occurring and how many of these types of proteins are there. And so Maureen has already introduced the fact that these C-type carbohydrate recognition domains occur in many different protein contexts. What I've shown on this slide, just for completeness, is the fact that the C-type lectin domains also occur in lots of different protein contexts, but these are the ones that we would not necessarily predict would bind to sugars. So we have to be careful because we're going to concentrate on the ones that actually do bind to the sugars. Within that group, we can make some interesting sort of comparisons. So first of all, they fall into, if we just look at the sequences of the sugar binding domains, the carbohydrate recognition domains, and we classify the proteins based upon those sequences, the classification that we get, where the most related sugar binding domains are each of these different ones, correspond exactly to the different structural groups. So that says that each of these structural groups evolved, and then the carbohydrate recognition domains within them sub-evolved in some different ways within the groups. And that can be very useful to us in understanding their sorts of functions. This slide also highlights the fact that all the ones with labels on them here are the ones that do bind sugars, and the ones with the green labels on them are the ones where there are structures, some of them from us and some of them from other groups. And you can see our structural understanding is pretty good at this point. We've got lots of examples, and those are structures where we have sugars bound to the binding site. So we know pretty much a lot about what the different ways that they can bind sugars. And so I won't go into a huge number of different details about this, but I do want to emphasize that two things about the evolution. One is something you've already heard about, which is the fact that these invertebrate organisms are very different to vertebrates. And this is true within the C-type lectin family. So you've already heard that it's different for different lectin families. But within the C-type lectin family, there are no examples of proteins that clearly existed in the progenitor and then have subsequently evolved. The domain existed, but there have been no proteins that otherwise are similar. But even within the vertebrates, there are some really quite recent swapping around, just as you've seen within the siglecs, that there is difference between mice and humans. That is also true in the C-type lectin family. There are examples of certain proteins that occur only in humans or only in humans and very closely related apes. There are quite a number that do exist widely across the animal kingdom, but there are also ones where there are now two versions in humans or multiple versions in mice. And there are even, the example that isn't shown here, one that has disappeared from mice but is found in most other species. And so it can be a little bit frustrating. In some cases, animal models can be very useful. In other cases, nature's already made the mouse knockout, and so we're stuck. There's not much we can learn by studying the mice. Of course, the binding sites aren't just as simple as the monosaccharides, and this is where they does start to differ significantly from the siglecs, because the binding sites can be more extended. And so we can get these quite lovely glycan array results where we put a protein, in this case dectin 2, it's a receptor found on macrophages that binds to yeast in particular. And if we put it on the lectin array and score what are the things, on the glycan array, and score what are the things that it binds to best, all of the things that it binds to best are high mannose oligosaccharides. Now, unfortunately, what's on the glycan array are mostly mammalian high mannose oligosaccharides, so they look like this. But what it tells us actually is that what it likes to bind to is these 1,2,1,2 linked mannose residues. And that's actually what's characteristic of the side branches on the mannans on the yeast. And so that becomes a very good indicator for a foreign type of glycan. You're not going to put a lot of these things on your own cell surface most of the time, but these will be quite abundant on the pathogen surface. And the binding site correspondingly has a mannose in the primary binding site, just like in mannose binding protein, but it also makes contacts with these other mannoses that are part of the chain. So the binding site is extended and there's kind of a secondary binding site over here. So we can gain specificity by extending the binding site and make it more of an oligosaccharide binding site. And we gain some affinity, although still not a huge amount. There's multiple ways of interacting with high mannose oligosaccharides. DC sign, the dendritic cell receptor, that's best known for binding the high mannose oligosaccharides on HIV, also has a binding site that likes to bind to high mannose oligosaccharides, but it actually tends to bind this branch structure out here, but it still binds the mannose in the primary binding site, and then the rest of the oligosaccharide is looped into this sort of cleft. So it's really quite, if you do a space filling model, you see how well it sort of fits in there. So it starts to look like a real binding site rather than one of these kind of crummy, very flimsy coordination binding sites. And it becomes a little more familiar, sort of biochemically speaking, to what we're used to thinking of as a binding site. The binding sites can also lead to other types of specificity for other sort of special marker glycans on mammalian cells. And so this Lewis X epitope, which has the terminal galactose and a terminal fucose residue sticking out, sort of packed against each other, is found only on certain types of proteins in mammals. And there is a receptor, the scavenger receptor C-type lectin, also known as galectin P1, has a sugar binding domain way out at the end of all this stuff, which specifically binds that trisaccharide motif. And that gives this actually the ability to take up Lewis X containing structures and do endocytosis. It's a receptor actually normally found on endothelial cells. This is transfected into fibroblasts. And it takes up, in this case, the Lewis X tagged neoglycoprotein. Naturally, what it can take up are proteins released from neutrophils. So the neutrophil granule proteins are tagged with this tag, and that means that once they're released, if they end up in the blood, they will get cleared very quickly by the endothelial cells. So they're released where the neutrophil is attacking at an inflammatory site, but if they get, you don't want them, these hydrolases that are being released from the neutrophils getting around, and so this tag is a way for the endothelium to recognize those proteins and clear them away. So we can end up with a binding site that's more specific by binding more sugars, more complicated sugars. And finally, we can end up with a binding site that ends up, interacts with things beyond the sugar. So the Minkel is another macrophage receptor that also binds sugars like mannose, but what its natural ligand seems to actually be a glucose residue, which is quite unusual in a glycoprotein. You wouldn't normally have glucose, but it does occur in certain glycolipids, in particular mycobacterial trehalose dimycolate. So the trehalose has two glucose residues. The glucose can bind in the primary binding site with a bit of a secondary binding site for the second glucose, but this is normally attached to this glycolipid structure, and the binding site is actually extended to allow interaction with the non-sugar part of the ligand. So you can also have specificity coming from these additional types of interactions. The other, of course, well understood example of that are the selectin ligands that Maureen mentioned, where you can bind, for instance, to sulfated residues in the ligand protein PSGL1. So you can have additional ways of attaching specificity, and those kinds of things are all summarized in this slide, which shows you that you can have the primary binding site, very simple recognition. You can extend that to looking at longer types of sugars, and you can extend it further by looking at additional things beyond just the sugar. And then finally, for recognition, we put together multiple carbohydrate recognition domains into oligomers. And so we can cluster the binding sites. And so we go beyond just looking at monosaccharides, oligosaccharides, and glycoconjugates to looking at actual surfaces of viruses and organisms. And just two examples of that. So the first of these is a protein that Maureen already alluded to, mannose binding protein. Sorry, I managed to hit the key there. Mannose binding protein consists, in the first instances, of trimers, which have 3 sugar binding sites. And there's a very specific characteristic of the way those binding sites are held together, which is that they're absolutely rigidly positioned. This is not like an antibody where you have FAB domains that are flexible. This is absolutely rigidly interacting here. And you can see the 3 binding sites there. And they are, first of all pointing in one direction. So they're pointing up here, so that if you attach an oligosaccharide at each of those positions, they'd each be pointing one way. So that's ideal for looking for a cell surface. And the interaction, the binding sites are 50 angstroms apart. Way too far apart for 2 sugars in one oligosaccharide to interact. So you can distinguish self from non-self by the density of the glycans. So you need these very dense arrays of sugars that you have on the surfaces of some of the microorganisms that are recognized by mannose binding protein when it initiates complement fixation. The other way you can recognize a pathogen surface is by making a more specific recognition site for a pathogen-specific glycan. So that's what DC Sign is doing when it recognizes the high mannose oligosaccharide on HIV, because that high mannose oligosaccharide is not going to be common on your surfaces, but will be more common on the virus surface. We don't completely understand why that is, but in general, viruses are much more likely to have high mannose oligosaccharides, possibly because they bypass the last part of the secretory pathway and get to the cell surface without fully processed glycans. But in this case, the oligosaccharides then have to fit into this binding site only in sort of one orientation. And in order to accommodate that, you do need to introduce flexibility between the binding sites. So now you have binding sites that can swivel around so that they can interact with the oligosaccharides on the surface, multiple oligosaccharides on the surface of the virus. In either case, you're making use of the multivalency to increase the affinity from the very weak affinities, millimolar affinities for the monosaccharides, and even for most oligosaccharides, not much better than millimolar. Now you can start to have quite tight binding to the surfaces. Okay, so what I've left myself time for is to tell you where we are and where we aren't in terms of understanding how does this recognition, all these different steps in the recognition, lead to biological outcomes. And it's quite good to have Matt talking to you beforehand, because you've got real sort of bona fide examples of things where we do understand something about the signaling. But I'll tell you some other examples where we may or may not be on the right track. So how do they lead to functions, and how can we exploit that? So one example that I can tell you about that is pretty well understood up to a point is this interaction of mannose binding protein with the surface of the microorganism, because we know what that does. Now, it doesn't involve signaling in cells. Mannose binding protein is a serum protein, as Maureen said. It's involved in initiating complement fixation, but that's still a matter of transmitting information. You have to tell the complement, this is a foreign cell surface, and activate complement on that surface so that the pathogen will be neutralized. And what we think that involves is, although these binding sites are rigidly adjacent to each other in the trimer, when you put together multiple trimers into the larger mannose binding protein oligomer, there is flexibility between the different arms. And that is very definitely true. We can see that in the microscope. And if the mannose binding protein associated serine proteases are attached here, they're going to sense the interaction of the arms with the cell surface. So when the cell surface binds to the trimers and locks them into position, that is likely enough to bring the proteases close to enough to each other that they can self-activate. And when they self-activate, then that initiates the complement cascade. So that's an example of where it's not the sugar binding to an individual CRD that's starting the signaling, and that's the key point. The actual interaction is too weak. It's can't drive anything, and in almost all cases, there's no change in the CRD when the binding occurs. There's no conformational response to the sugar. The conformational response comes to the cluster of sugars, to the organization of multiple sugars on the cell surface being sensed by the multiple binding sites. And I think that's a very useful paradigm for our way of thinking about the way sugar binding receptors can sense information and can initiate a response. Even though most of the receptors we're interested in are cell surface receptors, I think that's still a useful way of thinking about it. The other thing that's nice about mannose binding protein is we can make a very specific linkage to function through human mutations. And again, Maureen introduced this, but I want to emphasize that we really understand why this mutation does what it does. So these are the sugar binding domains. This is this long collagen-like domain. And these mutations are in the collagen-like domain. And these are types of mutations that are very well studied in other collagen-containing molecules. They will prevent the formation of the collagen triple helix. Just the one simple mutation of changing a glycine to something else will certainly prevent the ability of this molecule to assemble into its full trimeric state. And in fact, we know that's true. Once the folding starts at this end, goes as far as it can go, and then it can't finish assembling the rest of the molecule. And if it can't make this molecule, it can't come together into the higher oligomers, and it can't therefore do any of the complement fixation. And it's worse than that because it messes up. If there's only one polypeptide in the trimer that's wrong, that's enough to prevent the trimer from working. So the mutation becomes essentially dominant. And that's why it can be picked up in a disease-causing mutation, because infants with this mutation will not have this first line of innate immune response against pathogens because they won't have serum levels of mannose binding protein that can help them before they can make antibodies. So mannose binding protein kind of ticks all the boxes. We have the fact that we have a linkage to susceptibility, very clear population genetics information that says those mutations lead to susceptibility to particular types of infections. We can describe what the effect of the mutations are on the physical properties of the protein. It doesn't affect binding to sugar per se, but it affects the downstream effects, and that's enough to explain the disease. We have hints that there are going to be other such examples. The mannose receptor is very tightly genetically linked to susceptibility to mycobacterial infection in the case of leprosy. The exact mutation that's associated with that remains to be elucidated, but the implication is that some of the mutations certainly are in positions that we would expect to change the stability of the sugar binding domains. But we haven't filled in the rest of the boxes. DCCNR certainly has polymorphisms that can change the oligomeric state of the receptor, but linkage to there have been on and off reports of linkage to viral susceptibility. Longerin has actual mutations in the binding site that change the specificity, all of these in human populations. In this case, that they can cause differences in which ligands are interacted with. So we're hoping that this table will grow and we'll be able to map out additional sort of functions through all those different steps. And the final thing then to say is there is some hope from this, which is that we think we can start to understand some of the types of signaling interactions, thinking somewhat the same way as with the siglecs. But what I want to emphasize is that there would be two kinds of ways you could think about a ligand interacting with one of these receptors that contains two C-type lectins and one of these Sic kinase binding domains, very much like in the siglecs. One would be that there would be an interaction with an oligosaccharide ligand where two sugar binding domains within a dimer would somehow change the way the dimer behaves, and that would start signaling. For a lot of reasons, we now think that's probably not the likely way it's going to work, that the dimer, again, is preformed and almost certainly rigid in its orientation. We know what some of the dimers look like. What is going to happen is that the binding sites will often be able to bridge two dimers, bring the dimers together, and make one of these clusters that can then start the activation process. And we can see some examples of where we've seen oligosaccharides that can bridge between, not within a dimer, but between two dimers. So we're favoring that model. However, just to show the last slide, there is mileage in targeting the oligosaccharides. And so there are now, it's taken a lot of years. It was almost 20 years ago that Alna Lamb asked to speak to me after a glycoconjugate meeting for sort of vague reasons, which I now understand involved the fact that they were trying to target siRNA to liver in order to treat various diseases. And they now sell drugs based on that basis. They have a whole pipeline of drugs, several of which are actually in the clinic now, not just in clinical trials, but are approved, where they target a particular siRNA, in this case for acute hepatic porphyria, which involves, in all cases, shutting down overproduction of something in the liver. So you send an siRNA there and damp down production of your target, and the way they get it to the liver is to put a tag on it, which is a trisaccharide. Well, it's not a trisaccharide. It's 3 N-acetyl galactosamines that will target the asialoglycoprotein receptor on the liver cells. I don't think they quite know why they put the 3 there. They put the 3 there because we drew this picture of the asialoglycoprotein receptor with 3 subunits. In fact, I don't think this ligand can bind into those 3 binding sites in the trimer. But the fact is it works. They did a lot of pharmacokinetics and came up with a tag that very nicely targets their thing to the liver. So we'd like to get the understanding further, but even with our current understanding, there's useful things that are starting to come out of it. So I thought that's kind of neat that actually after all this time of studying the receptor, there are useful outcomes. And I'll stop there.
Thank you very much. Any question from the audience?
I have one.
Go ahead.
So what do you think about this idea that ER stress causes high mannose, and maybe that's somehow inducing ligands for these receptors? I mean, that's sort of breaking self, non-self, but is it happening for a reason, maybe?
Yeah, it's possible. It's hard for me to know. I still don't have a good fix on the density, whether you because again, one high monooligosaccharide getting to the cell surface isn't going to be enough. That's the whole point. You do have occasional high mannoses already. The targeting comes when you have the dense arrays in the high amounts. So I don't know whether it's going to be enough of an effect that the receptors would recognize it, but it's plausible that's on purpose to signal distress in some way.
Any other question? Let me ask one question. If we look at the human genome, if we search the homology, do the homology search using the human genome as a target, there are about 80 or so C-type lectin-like sequences, if I remember. But out of the only 30 or 31, according to Dr. Taylor's slide, are legitimate C-type lectins with the Glycan-binding function. Out of the rest, maybe about 50 genes, there are some exceptions like lectin-1, which binds to the beta-glucan, and also the siglecs-2, which binds to sialylated oligosaccharides. What would be your prediction that out of these 50 other CTLDs, how much of that could potentially bind glycans?
Yeah, I can't see any common additional sequence that gives me a basis for predicting additional ones. So it's kind of, given that the number of different sugar binding, the number of different protein folds in which you can build sugar binding domains, it's not clear to me that it's any more common in the C-type lectin-like domains than in immunoglobulin domains or any of the others. So, and there have been hints with a few of the others, but whenever we've tried to follow up on them, it's proven very difficult. So I wish I could have a bioinformatic approach that would help me to spot why do these other ones also bind sugar. So having the calcium is critical for our way of binding sugars, but not having the calcium certainly doesn't prove that it won't bind sugar. But it just it certainly reduces the likelihood statistically.
Yes. Another question, if we line up these 80 different or maybe more of the C-type lectin-like domains and with the legitimate C-type lectin domain CRDs, do the CRDs tend to cluster together?
Yeah, because in each of those groups, each of the sugar binding groups was distinct from the non-sugar binding groups.
I see.
Yeah. So historically, the loss of or gain or loss of sugar binding activity occurred in the whole group.
I see. So there is presumably one ancestor which acquired the binding to sugar and have.
Yeah. The only peculiarity to that is that we know that you can. What it takes to go from being a mannose binding to a galactose binding domain. And you have to change like 3 residues. And we know that has happened more than once in different families. But in terms of gaining sugar binding activity, that seems to have happened once.
Great. Any other question? Yes, Dr. Koo.
There's one question I always wonder, because whenever there is a virus coming out, people always say that it binds to C-type lectin. And indeed, it may not be. And in the case of COVID-1 and COVID-2, there are several reports on that. So what's your take-home message for people that is there any function for C-type lectins in, let's say, COVID-19?
Well, when I talked about the lectin array in the main meeting, I will not show any results for COVID, specifically because it binds everything. And the people who work on it, who generate this weren't surprised, because not just in the C-type lectin family, it just sticks to everything. And so you have to be very careful in interpreting the results. So individually, I'm sure it's true. You know, if you take your favorite C-type-lectin and COVID, they'll interact with each other. But you could take any other C-type-lectin and it would also bind. So at this point, I don't see a well-defined role.
Thank you very much. Is there any urgent question you want to ask? If not, let's thank Dr. Drickamer for outstanding talk. And also, let's thank all the speakers of this session. Thank you very much.